Hello students, today we are starting with the chapter cell cycle and cell division. So we all know that all living cells or all living organisms are composed of cells. Cell is the basic unit of life. Cell is a structural and functional unit of life. You might have studied in the chapter Cell, the unit of life. What is the importance of cell? What are the features of the cell? What are the different cell organelles present in the cell? But in this chapter, we are going to see cell cycle and cell division. So, all organisms are composed of cells. The cell could be in an organism, they could be a unicellular organisms that contain a single cell, or the multicellular organisms can contain many number of cells. Now, in the unicellular organism, it contains single cell, but it performs all the activities required for the growth and development of an organism. Whereas the multicellular organism also developed from a single cell. Now, the cell that goes on to form large organisms will possess two important characters, that is growth and reproduction. Growth and reproduction are characteristics of cells. So, these cells reproduce by dividing through cell division process, wherein the parental cell gives rise to the daughter cells. So, there is a cycle of events taking place from formation of one cell to two cells. As the theory states by Rudolf Virchow, that is omni cellula e cellulae. What does it mean? New cells are formed by the division of pre-existing cells. So, in order to form the new cells, the pre-existing cell must undergo cell division. So, in this chapter, we are going to see what all are the events that are taking place from division of one cell to the division of another cell. What are the factors that are affecting this cell division? What are the activities that are taking place in this cell division? Right? So, let us see what is cell cycle and what is cell division, and what are the activities that are going on in these two processes. Okay, in the cell cycle and cell division. Cell division is a part of cell cycle. Cell division is a part of cell cycle, but cell cycle, once an organism undergoes cell cycle, one complete cell cycle, then a parent cell will be divided into two daughter cells. So, all the events that are undergoing in order to divide a single cell into two different cells which are identical to the parent cell, it is known as cell cycle. So, let us see the definition of cell cycle. Cell cycle can be defined as sequence of events by which a cell grows, synthesizes various constituents, duplicates its genome, and divides into two daughter cells is called as cell cycle. It is a sequential order by which a cell duplicates its genome, synthesizes other constituents of the cell, and eventually divides into two daughter cells. Means all the activities, all the preparations that are required for the cell division, okay, everything is performed in cell cycle. So, the orderly manner of the events that are taking place in order to prepare a cell for the division purpose, in order to divide a parent cell into two daughter cell, all these events in a sequential manner that is going to take place is known as cell cycle. Now, it is mandatory to carry out all the events for the cell division purpose to take place in a sequential manner or in a orderly manner. Why? In order to produce the progeny cells or the daughter cells that carry intact genomes or without the loss of any genetic material. If the steps in the cell cycle is going to be randomly or in a haphazard manner, then the resulting progeny cells may contain certain extra genes or extra chromosomes that will lead to the disorder conditions. Therefore, all the events required for the cell growth and cell division must undergo in a sequential way or in a orderly manner. That is the reason it is termed as cell cycle. Once again, I'll define cell cycle. It is defined as sequence of events by which a cell duplicates its genome, synthesizes the constraints of the cell, such as if a parent cell has to divide into the daughter cell, it has to distribute its cell organelles, all the cell structures to the daughter cells. So how it will distribute? When they are already synthesized, then only it will be able to distribute. So synthesizing these constituents of the cell and eventually dividing these parent cell into two daughter cells is termed as cell cycle. Let us see some of the events contributing to cell cycle. What all are the events that are participating in the cell cycle for the cell division process? One is DNA replication, which is very important part of the cell cycle. Why? So, cell division is a part of cell cycle. When a cell is divided and said to be complete when it possesses the genetic material. So, the basis for the genetic material is DNA. Now, how the DNA can be distributed to two daughter cells? When it is divided or when it is replicated or when it is duplicated, then only it can be divided into two daughter cells. So, the first event or one of the most important event in the cell cycle is DNA replication for cell division purpose. And then this DNA replication in the cell cycle takes place at a particular specific stage in the cell cycle. And cell growth takes place in the cell cycle. Increase in the cell size as well as producing all the cell organelles is known as cell growth. Cell growth is a continuous process which is in terms of cytoplasmic increase. The nuclear content will not be duplicated in the cell cycle or distributed to the daughter cells, but only there is increase in the cytoplasmic ratio. Synthesizing the other constituents of the cell like subcellular components or the cell organelles, they are also once again synthesized in the cell cycle. Synthesizing proteins required for DNA replication. So all the important steps, all the preparatory steps that are required for the cell division, right? Everything is, cell division is also a part of cell cycle. So some of the preparatory steps for the cell division also involved in the cell cycle. These are also the events contributing to the cell cycle, such as DNA replication and also the replication of the cell organelles, that is synthesizing the constituents of the cell, and replication of chromosomes. Replication of chromosomes will not take place in the interphase, but it takes place in the cell division, right? So replication of chromosomes, distribution of chromosomes to daughter nuclei, that is during the cell division process. In the cell division process, there is no preparation, but there is only the distribution, right? So this is also one of the feature of the cell cycle. Distribution of chromosomes to the daughter cells in a complex series of events during cell division. So in single stage, the chromosome division is not going to take place. Once again, the cell division has two different phases, and each phase has once again sub stages or sub phases, which we will be seeing in the further video. So all these events that are contributing to the cell cycle must be in a sequential order. After synthesizing of DNA, then only the division of DNA can takes place, and then only the distribution of DNA to the daughter cells can takes place. So all these are in a sequential order. Now all these events will pass from one phase to the another phase, means from one stage to the another stage. So these events are under genetic control. They are under genetic control. How the passing of events or the transformation from one stage to the next stage is going to be controlled. It is controlled through a specific types of proteins known as cyclins. There are specific types of proteins known as cyclins and cyclin dependent kinases. These are enzymes. Cyclins are proteins and cyclin dependent kinases are enzymes. Together they help in transforming from one stage to the another stage. So in the further video you'll be knowing what are the different stages that are involved in the cell cycle and how each stage is transformed to the another stage. So each stage will be passed on to the another stage, means how a cell will know now the one phase is completed, now the cell has to enter into the next phase. That is all because of the proteins known as cyclins and cyclin dependent kinases. What are cyclins? Cyclins are once again a set of proteins which regulates the cell cycle by binding to the cyclin dependent kinases. Now what are these cyclin dependent kinases, or what are kinases? Kinases are the enzyme which add the phosphate group to a particular compound. Okay, kinases are the enzymes that are involved in the addition of phosphate group to a compound, and the process of adding phosphate group to the compound is known as phosphorylation. It is known as phosphorylation. So therefore the cyclins and the cyclin dependent kinases will result in phosphorylation process. So what will happen? The activated CDKs, means cyclin dependent kinases, will either initiate or inhibit the activities in each stage by phosphorylating the proteins of specific stages. When it is phosphorylated, such as adding phosphate group, so adenosine diphosphate is present. If a phosphate group is added to this adenosine diphosphate, it will become adenosine triphosphate, which is one of the form of the energy. So once the energy is being transformed, it will activate to produce different activities in that particular stage. So therefore the events in the cell cycle are under genetic control, and therefore the particular proteins involved are cyclins and cyclin dependent kinases. Now we will see the duration of the cell that is taken to produce two daughter cells from a single parent cell. The cell has to undergo one complete cell cycle. So the duration required to complete one cell cycle by an organism is going to be known as generation time. It is known as generation time. So this generation time can be defined as it is a period required to complete one cell cycle, that is from beginning of one cell division to the beginning of the next cell division. Not till the end of the same division, but till the next cell division begins. That particular time period or the time interval is known as generation time. This generation time is going to vary from one organism to another organism, right? So if you see the generation time, or the time taken by the eukaryotic human cell to complete its one complete cell cycle, it takes 24 hours. So every 24 hours a cell undergoes cell division. Once again in between it doesn't, but it is going to divide in every 24 hours. This time is going to vary from human cells or from one organism to the another organism. If we see the generation time in yeast cells, the cells are going to double. at every 90 minutes. So in 90 minutes the yeast cell will complete one complete cell cycle. It is going to complete, means from the beginning of one cell division, so it is going to produce two daughter cells, and then it is going to enter into the next cell division. The two daughter cells whatever were produced will further undergo cell division after 90 minutes, right? So 90 minutes is the generation time for yeast cells. Similarly for E. coli it is 20 minutes, and Staphylococcus aureus it is 30 minutes. So therefore most of the experiments are performed in these microorganisms when compared with the human cells, because these cells are going to produce the population, okay, at a very early stage, because their generation time or the doubling time means it the time taken to double from one single cell to the next cell, that is nothing but the period taken from one cell division to the next cell division. So that is known as generation time. Now let us see, cell growth and cell division are the part of the cell cycle. So what is cell division? What do you mean by cell division? So we all have been knowing that it is a process by which a mature cell divides and forms two daughter cells each time they divide. Or it is a process by which parent cell synthesizes its components, divides and forms two daughter cells. Yes or no? So, in the cell cycle, there is a period known as cell growth. During this cell growth period, the cell will synthesize all the required components that are required for the division, and ultimately it divides and forms two daughter cells by cell division process. Cell which undergoes division is called a mother cell or a parent cell. The cell that undergoes cell division is called a mother cell, or it is also known as a parent cell. And the newly formed cells means, as Rudolf Virchow says, new cells are produced from the pre-existing cell. So the pre-existing cell is known as a mother cell or parent cell, and the newly formed cells are known as daughter cells. Now cell division is a process by which the cell will synthesize its components and divides and forms two daughter cells. Cell reproduction is formation of new daughter cells from parent cells through cell division. It is formation of new daughter cells from parent cells through cell division. Okay. Now the cell division could be the means of reproduction in some types of organisms, such as in case of unicellular organisms. In unicellular organism, they are single cell organisms. So reproduction is producing the offsprings or producing the progeny cells. So in case of unicellular organisms, the reproduction by which the mother cell produces two or more new cells. So it is a means of reproduction. When in case of multicellular organisms, the new individual develops from a single cell. A new individual is going to develop from a single cell. How? A haploid set of chromosomes is taken from a parental side, and another haploid set of chromosomes are taken from a maternal side. Together it will fuse to form a zygote, which is a single cell. From this zygote, it is going to produce another cells. Okay, the repeated cell division of this zygote is going to take place, and many millions of cells are formed in order to form a multicellular organism which is having a organ system level of organization. Yes or no? We have an organ system. We have organs. So how these organs are produced? Initially, there is a single cell formed known as zygote. This zygote is going to undergo repeated cell division. Cell differentiation is going to take place, which is giving rise to tissues, and number of tissues together is going to form organs. Okay. So in multicellular organisms, a new individual is going to develop from a single cell. So therefore cell division is considered as it is a central to life of all cells. Right? So now let us see in detail the cell cycle. What do you mean by cell cycle? Cell cycle. Now we have seen what is cell cycle. It is a sequential manner of events that is going to take place in order for the cell to grow and to reproduce. So all the events that are required are performed in a sequential order for the cell growth and cell division purpose is known as cell cycle. So the cell cycle is divided into two phases. First phase it is known as interphase and another phase is known as M phase. One phase is known as interphase and another phase is known as M phase. So these are the two phases involved in cell cycle. The cell cycle involves, as I said, cell growth and cell division processes. Interphase is the cell growth purpose. M phase is a cell division process. Getting? Interphase is a cell growth. So interphase is also known as I phase or it is also known as long undividing phases. But M phase is known as mitotic phase where the division of nucleus is going to take place. Whereas in interphase, the cell is not going to divide but the cell is only going to grow, increase in size, not in number. Cell is not going to increase in number but only it is going to increase in size. That is, cell growth is going to take place in interphase. So this phase is known as long undividing phase. It is known as long undividing phase or it is also called as resting phase or it is also called as not dividing phase. Getting? Interphase is also known as resting phase or undividing phase. Long undividing phase. Why long undividing phase? In a complete cell cycle, the cell going to spend long term or long duration in interphase. Okay, so this phase is between two successive M phases or between two successive cell divisions. Interphase is the phase between two cell divisions. Between one division and another division there is a interphase. Means cell growth phase is going to take place. After that only the cell division is going to take place. So it is a phase between two successive M phases or they are also known as cell division phases or they are also known as mitotic phases. And it is a period that is between the end of one cell division to the beginning of the next cell division. As I said, the cell is going to spend 95% of duration of cell cycle in interphase. Why it is also called as resting phase? Though the cell is growing, it is also called as resting phase because there is no cell division taking place. Though it is called the resting phase, it is highly metabolically active in which the cell prepares itself for the next cell division and the cell prepares for division by undergoing both cell growth and DNA replication in an orderly manner. But here there is no division of nucleus or the parent cell is not divided into daughter cell but only the cell growth and the DNA replication is going to take place in an orderly manner. Whereas M phase is a mitosis phase and this phase is further divided into subphases. Okay, so let us see what are the different phases of interphase or the interphase is divided into different phases, three different phases. 1 G1 phase, 2 S phase, 3 G2 phase. The interphase is subdivided into three phases. One is G1 phase, G2 phase and S phase. All these three are cell growth and DNA replication phases but there is no cell division. In this phase there is no cell division. So what is this G1 phase constitutes? or what are the activities that are performed in the G1 phase? G1 phase is also known as post-mitotic phase, and it is known as pre-synthetic phase, or it is known as first growth phase, or it is known as gap 1 phase. Getting? G1 phase is also known as post-mitotic phase, pre-synthetic stage, first growth phase, and gap 1 phase. In the G1 phase, it is the interval between the mitosis and S phase of interphase. Because after this interphase, next phase is going to be mitotic phase, or it is a cell division phase. And after this G1 phase of the interphase, next stage is the S phase, or it is the synthesis phase. So the interval between the mitosis and S phase, so it is known as post-mitotic phase, or it is known as pre-synthetic phase. Now the cells are in high metabolic rate and grows continuously. Cells are in very high metabolic rate. Why? They start producing different biochemicals that are required for the DNA replication in the next phase, and therefore they grows continuously. In this phase, the cell can be observed to increase in its size because lot many components have been produced within the cell size is going to be increased. Apart from the increase in the cell size, okay, there is also the increase in the nucleus size, but not to the higher extent. Very small size increased in the nucleus. That is because of the synthesis of ribosomes and RNA. And there is no DNA replication in this phase, and there is no change in the amount of DNA. Because of this reason only, there is no much increase in the size of the nucleus also. So here the DNA replication is not going to take place in the G1 phase. In G1 phase, the metabolic rate is high and cells grows continuously but does not replicate its DNA. So in this phase, the biochemicals such as RNA, ribosomes, proteins, lipids, and also cell organelles such as Golgi body structures for the next stage are being synthesized in the G1 stage. In G1 stage or in the G1 phase, a cell can take one of the two pathways, or it has a diversion. So it has an option to take in many or to continue in any one of the next phases. In G1 phase, a cell can make a decision either to enter one of the two phases. So what are these two pathways? That is G0 phase and S phase. A cell in the G1 phase can decide either to enter G0 phase or to enter S phase. Now what is this G0 phase? Usually in the cells that are dividing for the long term, a cell from the G1 phase will enter into the next phase, that is S phase. But the cells which are not wishing to continue the cell cycle, or which are not long-term dividing cells, or which do not exhibit division, or the reserve cells which wants to continue the cell cycle in upcoming time, or which has not received any signal through cyclins for further cell division purpose, all these cells will exit the G1 phase and enter into the next phase known as G0 phase, or it is known as quiescent phase. It is known as G0 phase, or it is known as quiescent phase. In this phase, the cell proliferation is not going to take place, but the cells are metabolically active. The cells are metabolically active, but they enter an inactive stage because there is no cell division is going to take place. Getting? The cells that do not exhibit division, or the reserve cells, or which can join the cell cycle on receiving signal through cyclin proteins, or divide occasionally to replace if there is any accident because of which the cells are lost, okay, or due to the cell death. These cells will exit the G1 phase and they enter the G0 phase or quiescent phase, where in this phase the cyclin proteins are not controlling the cell division, that is, there is an absence of the cycling proteins that control the cell cycle. So here there is no synthesis of the cell organelles taking place, and there is no synthesis of the DNA is going to take place, but they are metabolically active, and unless they get a signal from the cyclin proteins to enter to the further cell division process. So some of the examples are cells that are undergoing or that are exiting the G1 phase and entering the G0 phase or the quiescent phase are heart cells, nerve cells, and erythrocytes. They do not divide further and they remain in the G1 phase for long term or permanently. But in case of liver cells, these cells are once again continuing the pathway. They can resume to G1 phase or they enter into the S phase in response to certain stimulus, that is in certain chemicals such as cyclin proteins. When there are the cyclin proteins that control the flow from one stage to another stage are formed, then automatically these liver cells will enter into the cell cycle. So G1 phase is one of the phase of the interphase. Okay, and from the G1 phase a cell has an option to enter either the G0 phase or the S phase, and those cells which are not dividing or which are not long term dividing or which are not continuously dividing and which are lost due to accident or which divide occasionally will enter into the inactive stage, which is known as quiescent stage or G0 stage. Next stage in interphase is S phase. It is S phase. S phase is also called as synthesis phase. It is also called as synthesis phase. In this phase, the DNA replication is going to take place, or it is a period during which the DNA synthesis or replication is going to take place for the cell division purpose. For the cell division purpose. So, the DNA replication is going to take place in the nucleus of the cell. Yes or no? You might have studied in the cell the unit of life, the composition of the nucleus, the structure of the nucleus, and the nucleus contain the DNA in the form of chromatin. Now, this chromatin undergoes replication, that is known as DNA replication is going to take place in the S phase, and that too in the nucleus. And there are centriole duplication that is going to take place in the cytoplasm. You have also studied the cytoskeletal structure of the cell, that are one of the component is centrioles, and these centrioles are required for the next cell division purpose. These centrioles are also duplicated in the cytoplasm. The important feature of the S phase is synthesis phase. So in this phase the DNA replication is going to take place in the nucleus, and in case of animal cells the DNA the centrioles are duplicated in the cytoplasm, because the centrioles are absent in case of plant cells. And there is increase in the amount of DNA in the nucleus of the cell in this S phase, because the DNA is replicated and it is doubled, so there is increase in the amount of DNA content but not in the number of chromosomes. Getting, students? The number of chromosomes are not increased or doubled, but only the content of the DNA is increased. For example, in diploid set, okay, in the cell, if the diploid set means two sets of chromosomes are present, so the number of chromosomes can be denoted as 2n in case of diploid set. Okay, this chromosome number will remain 2n only the amount is doubled from 2c to 4c. The number of chromatin threads are going to be increased but not the chromosomes. Okay, so in the diploid set 2n only will be present, but if the content is 2c, then it will be doubled to 4c. In case of haploid set, if the content is 1c, it will be increased to 2c. Okay, but the chromosome number in the haploid set will remain n, not the 2n, and in case of diploid set, it is going to be 2n, not the 4n. Clear? There is no increase in the chromosome number, but only there is increase in the content of the DNA. And in the S phase, another important feature is synthesis of histone proteins and non-histone proteins is going to take place from the precursors in the cell. Okay, the histone proteins and non-histone proteins, these are the proteins. Proteins are the important biomolecules of a living cell, and these proteins are made up of amino acids, and especially the histone proteins are made up of two types of amino acids, lysine and arginine. These are the basic amino acids which make up the histone proteins, and these proteins are very much required for the formation of chromosomes in the nucleus. Clear? So, the important features in the S phase is the division of or the duplication of the DNA in the nucleus, and the duplication of centrioles in the cytoplasm, and the synthesis of histone proteins and non-histone proteins from their precursors is going to take place in the S phase. After the S phase, a cell is going to enter into the G2 phase of interphase. It is going to enter G2 phase of interphase. G2 phase is also known as gap2 phase, or it is known as second growth phase, or it is known as pre-mitotic phase or post-synthetic phase. Now it is vice versa of the G1 phase. G1 phase was post-mitotic phase and pre-synthetic phase, because after mitosis a cell is going to enter the interphase, and in the interphase the first phase is G1 phase. So it is pre-mitotic phase, post-mitotic phase. Getting? After the mitosis, the next phase is gap1. So it is post-mitotic. Now G2 phase is pre-mitotic phase. After the G2 phase, a cell is going to enter the mitosis. So it is a pre-mitotic phase, or it is known as post-synthetic phase. Why post-synthetic? After the S phase it is going to take place. So it is known as post-synthetic phase. So it is the gap between the DNA synthesis and the next cell division. Once again, it is similar to the G1 phase. In the G1 phase or in the gap1 phase, the cell growth was taking place, and that cell growth is going to continue in the G2 phase or in the gap2 phase. There is increase in the cell size as well as nucleus size also increases. Now it is at a very nucleus size is increased to a very higher extent when compared with the G1 phase, because in the S phase the DNA replication has taken place, the amount of DNA has been doubled, so therefore the size of the nucleus is also increased in the G2 phase. As well as in the G2 phase, the types of biochemicals, the different types of biochemicals and the different types of RNA such as mRNA, rRNA, tRNA and other proteins that are required for the cell division purpose are synthesized in the G2 phase. As well as the mitochondria and the plastids and other cell organelles duplication is also going to take place in the cytoplasm in gap2 phase. So in this phase, the preparations are made for the next cell division or mitosis. In gap1 phase, the preparations are made for the DNA synthesis or the S phase, but in G2 phase, the preparations are made for the gap2 phase, mitosis phase or the next cell division phase. Right? So the cell growth is going to continue here, but apart from the cell growth, it is also going to once again produce the biochemical proteins as gap1 phase. Okay, and here once again the duplication of mitochondria, plastids and other organelles are going to take place, and microtubules are going to initiate the mitotic spindles. It is going to initiate the mitotic spindles, okay, are synthesized. Mitotic spindle proteins are synthesized in the gap2 phase. So these are the different features that are taking place in the G2 phase. So this is the diagram that represents a complete cell cycle of an organism. It has a complete cell cycle of an organism. So here G1, S, G2 and G0 phase has been shown. A cell will start its cell cycle from the interphase. Means before the cell division, a cell has to pass through the phase known as interphase. Interphase is divided into three phases, G1, S and G2 phase. And here the G0 phase is present. The undividing cells from the G1 phase will exit and go to G0 phase. But once again, if the G0 phase, the cells in the G0 phase receives the signals to continue with the cell cycle, then they resume back to the G1 phase and once again the cell cycle is going to continue. So what will happen in G1 phase? There is biochemical synthesis is going to take place in the G1 phase and the cell growth is going to continue, and certain types of ribosomes and other proteins are synthesized in the G1 phase and cell organelles are synthesized in the G1 phase. It is a preparatory stage for the S phase. So in the S phase, what happens? The DNA replication is going to take place in the nucleus. Centriole duplication is going to take place in the cytoplasm. Apart from the duplication, the histone proteins and non-histone proteins are synthesized from their precursors in the S phase. So S phase is known as synthesis phase, and here the amount of DNA is going to be doubled from 2C to 4C, but the chromosome number is going to remain same. It is not going to be doubled in the S phase, because when the chromosome number will increase only when it undergoes cell division process. So after S phase, the cell is going to enter to the G2 phase of the interphase. So in G2 phase, as gap1 phase, it is second growth phase or gap2 phase, where once again the cell growth is going to takes place and cell size is going to be increased. In this case, once again the active metabolic activities are going to taken place and biochemicals such as proteins are synthesized and types of RNAs are synthesized in preparation for mitosis. Okay, here the cell size increases as well as the nucleus size is also going to be increased, and mitochondria and plastids are going to be duplicated in the G2 phase. Right? After this phase, now the G1S and G2 phase constitute the interphase. After G2 phase, the cell enters the M phase. In M phase, once again there are different substages are going to take place, and this M phase has karyokinesis and cytokinesis. Karyokinesis involves prophase, metaphase, anaphase, telophase, and then it involves cytokinesis, division of the cell into two cells, that is division of cytoplasm is going to take place, that is cytokinesis. Karyokinesis, division of nucleus, but still two nuclei will be present in the same single cell, whereas in cytokinesis two daughter cells are formed here. Can you see this newly formed cell is beginning at the G1 phase in the interphase. Okay, this newly formed cell is beginning at the G1 phase of the interphase, and then after it is completing the M phase, it is resulted into two daughter cells. So all the events that are taking place from G1 phase till the formation of the two daughter cells after the cytokinesis, together all the events are in the sequential order, and which is known as cell cycle. And this cell cycle is differentiated into interphase and M phase. Now all these three is interphase. This is M phase, or it is a mitotic phase. Now G1, S, G2 and G0 are the cell growth, and this M phase cell division is a cell division phase. Both together will form a cell cycle. So in the next class we will see what is M phase and what are the sub stages of the M phase in detail.